Hello everyone, my name is Marazzano Colon, and today I'll be talking to you all about how nitrogen-fixing symbioses and bryophytes show differential colonization amounts depending on the cyanobacterial strain. And so, where's the clicker? Right. So, first and foremost, it is important to know that nitrogen is an essential nutrient for all plants, but it is often not found in forms which can be readily metabolized by the plant. And that's funny because it composes 78% of the Earth's atmosphere, but in a form called dinitrogen. And dinitrogen has a triple bond which is very difficult for organisms to break. Some organisms undergo a process called nitrogen fixation, which is the process of taking that triple bond, breaking it, and then converting the nitrogen into ammonia and other combined nitrogen sources which can be readily metabolized by the plant. An organism that can actually conduct it, nitrogen fixation, but industrially are humans via the Haber-Bosch process. And the only known organisms that conduct it biologically are prokaryotes via the only known family of enzymes called nitrogenases. And what nitrogenases do is that they convert atmospheric nitrogen into ammonia and other compounds that can be used. And so one such organism that can fix nitrogen and uses that can use a nitrogenase are cyanobacteria. And so cyanobacteria are a photosynthetic ancient phylum of nitrogen-fixing prokaryotes. And they have the ability to form heterocysts, which I've outlined here. And so heterocysts form an anaerobic environment that prevents oxygen from permanently degrading the nitrogenase enzyme. And so this ability to fix nitrogen allows the cyanobacteria to form symbiosis with plants, generally cyanobacteria from the genus Nostoc. And so I wanted to give you guys an idea of what this symbiosis looks like throughout the plant kingdom. And so first here, we have the angiosperm genus of plants called Gunnera. And so the cyanobacteria infect the mucus glands inside of the root stem. And then there are gymnosperms and cycads, which the cyanobacteria infect the mid-root cortex, which you can see here. And then there are the genus of aquatic ferns called Azolla, where the cyanobacteria live in the underside of the leaf and a leaf cavity filled with air. And then there are bryophytes. And in bryophytes, the cyanobacteria live inside of the mass of the plant and specialized mucus cavities. And so, what are bryophytes? I just used this term. I had no idea what they were when I walked into this building. I don't expect you guys to either. And so bryophytes is a formal classification of three types of plants: hornworts, mosses, which most of us know and have seen, and liverworts. And so the unifying theme of these type of plants is that they are generally small, seed-free, and that they are non-vascular. And on top of all this, they actually all have the main stage of their life form of their life is in the haploid stage. And that's in contrary to other plants, which all have diploids like humans. And so each type of bryophyte actually has species within it that forms Cyanobacteria symbiosis. But only two genera of liverworts and all species of hornworts form symbioses, where the cyanobacteria lives within the plant. And that is the topic of my experiment today. And so this is what we know about the symbiosis so far. So if I go to the same place around the same time of year, I will generally find the same cyanobacteria and the same host. And so this means that there is a consistency over space and time of the bryophyte cyanobacteria symbiosis. And the next thing that we know is it was found in a study that in one specific bryophyte species that there are varying colonization amounts of the cyanobacteria in these same mucus cavities. And also that they found in that one bryophyte species is that the cyanobacteria strains actually compete for colonization space inside of the bryophyte. And so the things that I'll be addressing specifically in my experiment are, do cyanobacteria actually show different colonization amounts throughout different bryophyte species? And also, previous reconstitution experiments, reconstitution meaning the reestablishment of a symbiosis between two known symbiosis partners. And in such studies, they were conducted in liquid culture. And this is not indicative of the natural environment of the Cyanobacteria symbiosis, because bryophytes live on land, as you see a lot of them there, like mosses. And so in our experiment, we wanted to see if we could use a solid media that mimics soil. And in that medium, in the lab, can we reconstitute the cyanobacteria bryophyte symbiosis? And so in order to do this, we, of course, designed an experiment. So 4 bryophyte species, 3 hornworts, and one of the only species of liverworts that can actually form the cyanobacteria symbiosis. And then we treated each 4 of these hosts with the cyanobacteria treatment. The control, which means no cyanobacteria, and then we treated them separately with 2 species of cyanobacteria. One cyanobacteria, Nostoc punctiforme, from a cycad symbiosis, and the other species was unidentified Nostoc spa from a hornwort symbiosis. And so we tested all possible combinations of 1 bryophyte and 1 cyanobacteria. And so what we did is that we put the bryophyte host in each corner, so these are all the same type, and inside of that, this is called the microcosm. It is filled with vermiculite that is sterilized, and also we put a nutrient media for the plant called BCD, but it lacks nitrates and other nitrogen compounds. And this is because nitrogen deficient conditions are necessary in order to facilitate the bryophyte cyanobacteria symbiosis. And then we inoculated the microcosm dead in the center so that it was equidistant from all bryophytes. And so we collected growth data in 2 ways. First, we simply measured the weight of the plant as we put it into the microcosm at the beginning of the experiment and at the end of the experiment. And then we also decided to look at the surface area of the plants by taking a photo of the microcosms in the beginning and end of the experiment. And then we calculated the percent change throughout the entire experiment. So percent change of the before and after. And then we decided to measure cyanobacteria presence inside the plant by counting the amount of colonies per unit area. And so we simply did this by taking a subsection of the plant, putting it on the slide, counting how many colonies there were inside of that subsection, and then standardizing those colony numbers against the subsection area, and which would give us the colonies per centimeter squared of the bryophyte tissue. And so here's some data, specifically data of growth, but by percent area growth. And so here we have the 4 bryophyte species, 2 anthoceros, the Blasia pusilla, and the Dendrocero species. And that if we look within them, you can see that there are white boxes, which means plants treated with control, no cyanobacteria, brown Nostoc punctiforme, green the Nostoc spa. And so if we look at the data, we can see that all of the plants of the same species seem to grow by about the same amount. But within the plant, there is not much variation in growth. And so when we look at growth data considering weight, percent weight change over the experiment, we can see again, control, white, Nostoc punctiforme, brown, green, the Nostoc spa, that the plants of the same species all again grew by about the same amount. And if we look again, there's not much variance in considering the cyanobacteria treatments. However, we think that we would see more variance, especially between the control and the cyanobacteria treated bryophytes, if the experiment took place for a longer time to significantly affect plant growth. And so this is where we get some interesting data. And so here we have colonies per centimeter squared of plant mass or plant tissue. And so I have here removed the controls, so there's no controls because they all had 0 colonies per centimeter squared. And then we looked at we compared the 2 cyanobacteria treatments. And so if we look here, again we have the 4 species. And if we look within species, we can compare the cyanobacteria treatments. And then we can see that the Nostoc spa actually demonstrates higher growth in each bryophyte species. And that, in contrast, the Nostoc punctiforme generally shows not much colonization in all of the plants. And we believe that this is because the Nostoc punctiforme strain from Aggnnura symbiosis is just a poor colonizer, at least in bryophytes. And this is further supported by the fact when we looked at the colonization amounts of each treatment, irregardless of the strain in which the bryophytes were, the strains of the cyanobacteria which the plants, when we looked at the total area, when we looked at the total colonization amounts, irrespective of the cyanobacteria treatment, oh my God, there was a significant difference in the total colonization amounts of the cyanobacteria treatments, irrespective of whichever plant they were in. Simple, right? And so, in summary, we were able to successfully reconstitute all combinations of bryophyte and cyanobacteria. And this proves the efficacy of using vermiculite media, which mimics soil and future reconstitution experiments. And next, we also displayed that Nostoc showed greater colonization amounts in comparison to the Nostoc punctiforme, which generally showed little to no colonization in this bryophyte species. And then we also showed that there was no differences in the growth between treatments. We think that this is because of the time frame of the experiment. And so we have shown that the cyanobacteria strains show differential colonization amounts in different bryophyte host species, but we want to look at the bread and butter of the symbiosis, specifically looking at how much nitrogen is fixed by the cyanobacteria and how much of this nitrogen is actually incorporated into the plant biomass. And next, we want to get cyanobacteria from many different sources in order to look at the specificity of the plant cyanobacteria symbiosis. Another thing we wanted to look at is the cyanobacteria competition. And so previously, we've seen in that one bryophyte species that I talked about earlier is that there's competition between the cyanobacteria strains. And it'd be very interesting to see how that competition varies between each different bryophyte species. Another thing that we want to look at is growth rates of the plant throughout the experiment. And this is because we're interested to see if the symbiosis actually affects the plant in a temporal manner. And last but not least, you want to affect varying growth conditions in order to see if that affects aspects of the symbiosis. And so I want to acknowledge and thank every individual in my lab, which I was in the Lee lab, and my mentor, Jessica Nelson, for being with me in every step of the way, Duke for supporting my academic pursuits in my home university, the USDA and the NSF for funding, and Cornell and BTI for hosting me. Thank you, and I have time for questions. Boop. Yeah. Yeah. Oh, that is something that I should have mentioned in the presentation, that these are all in the wild. We have confirmed that they actually form the symbioses with the cyanobacteria. And actually, the Dendrocero species was actually the first time that we know about where it was actually reconstituted in the lab. Oh, yeah, they were grown in xenic, so without, the only organism inside the culture which I used was the bryophyte species. And so there's ways to get this bryophyte without, similar to bacteria culture, you just continue to grow different tissue samples until, there's no contaminants. And so I think, Josh, you had a question?
Yes. So the cyanobacteria that didn't colonize very well,
Mm-hmm.
did you notice any, I guess you probably didn't do this, but do you think it would be interesting to look at the genetic differences between the bacteria that successfully colonized versus the standard population that you dumped in the...
Yeah, I mean, most definitely. First, we wanted to get more cyanobacteria so that we could get more interesting loci. And then we can look at those loci, and I didn't mention it for time's sake, but to hopefully integrate cyanobacteria symbiosis and actually crop plant in order to further alleviate as everyone else mentions in their presentation, the future population growth. Yeah, so, yes?
So what are the cyanobacteria getting from the plant? Is it simply carbon?
Okay, so the cyanobacteria, they fix nitrogen which I've stated. And in exchange for the nitrogen, the bryophytes gives the cyanobacteria combined carbon. No. Or as far as we know. That's, it could be more complicated or than that, so. Okay, I guess that's it.